We're going to be talking today about our classification systems, how scientists are taking the time to actually find those particular characteristics of different types of organisms that are similar and put them together in groups so that we can talk about these particular groups and these generalities that certain organisms will have and use this information to understand how the rest of the organisms are working. So that's these classification systems. We have 3 different reasons for actually classifying organisms. Number 1, to help identify relationships between different kinds of organisms. An example of those relationships would be, what is that relationship? What connects something like a sea anemone and a jelly together? That's one of those relationships. And then we want to have scientists be required to clearly identify those key characteristics of different organisms. In other words, we want them to take the time to actually examine and observe those characteristics so that we know exactly what it is that we're talking about. And the third and final one is to avoid confusion. We have lots and lots and lots of different languages, from English to Russian to Chinese, Mandarin, Cantonese, huge amounts. And so we want to make sure that we're being very clear as to exactly what types of organisms that we're talking about. Now, in 1758, Carolus Linnaeus, a scientist studying plants specifically, was kind of confounded by a tremendous amount of information. And he wanted to organize the different organisms that he was looking at. And so he came up with his own Linnaean classification system. He used this classification system to organize organisms from a general, where there's a whole bunch of organisms within a particular group, to a very specific, where there's just a few, if not only one organism in that particular grouping. And that was the beginning of this scientific classification that we actually use today. He divided his organisms to groups that he called taxa, or taxon for short, for singular. And these divisions, he ended up coming up with 7 different divisions. Now, like I mentioned before, one of those reasons for classifying these organisms is because we have lots of different languages. Now, it's hard to see this, but here's a list of just a few of the languages that are out there in the world today, and the corresponding word for a dog. And when you're looking at some of these, not that a lot of you speak Armenian, but there's the shun. If you said that word in English, there might be a little bit of confusion. So we want to make sure that we kind of apply a particular terminology to all of these. Now, when you look at some of these particular names for a dog, what you'll come to understand is that a common name like dog isn't very accurate. Because what is it exactly that we're talking about? Because a dog could potentially be applied to many different species. We want to be just a little bit more accurate with our classifications. So over the last several centuries, scientists have come up with a particular naming system. It's called the binomial nomenclature. And binomial, bi meaning 2, nomial is names. And then nomenclature, again, that nomen is a name, and clature is a Greek for system. So this is actually the names of 2 parts naming system. And the goal of this particular system, one of these rules and basic principles, is to create a particular system where 2 things happen. Number 1, any single kind of organism has one and only one correct name. Let me say that again. Any single kind of organism has one and only one correct name. And number 2, no 2 organisms bear the same name. No 2 organisms bear the same name. For example, this picture here, you might be familiar with. This is a dolphin. So is this. These are both dolphins. So that's a problem when we start looking at things like those common names. And we say, oh, here's this organism is the dolphin. What is exactly the organism that we're talking about? We're talking about a dolphin mammal, or are we talking about the dolphin fish? This dolphin fish actually has several other names. A dorado, or like some of you might be familiar with, mahi-mahi. Mahi-mahi is a dorado, is a dolphin fish, which is very different than one of these. So we have these 6 basic principles that this binomial nomenclature actually goes by. The first one is that organisms from botanical, plants, zoological, organisms, and bacteriological, bacteria, they're independent of one another. So it is possible to have names that are similar in plants and animals and bacteria. Number two, a taxon, that is the group, can have one and only one correct name. I mentioned that earlier. No two genera or species. Genre is it's Genera. No two genera or species can have the same name. So they're very specific names. The scientific names, that is what we call our genus and species names, are treated as Latin, regardless of where language they came from. So we have to what we call Latinize those particular names. The correct or valid name comes from whoever publishes that name first. So if there's two groups or two individuals that have discovered the same species, they're doing the research to make sure that it is a new species. Whoever publishes it first and gets their name out there, whatever they're calling it, then that's the correct name. And then for the order, what we call the superfamily and below, the names must be based on what we call a type specimen. That is, we have to have an example in a museum. And there's several museums around the world that hold these particular example organisms. The American Museum of Natural History, one of the Smithsonian institutions in Washington, D.C., has huge, enormous rooms just filled with preserved organisms that are the type species. So it's based on, for example, a type that we have captured of this Dorado mahi-mahi, or this dolphin fish. So the example of every organism has to be somewhere in a museum. Now the classification system that we have, we'll talk about this here. It's organized, again, just like Linnaeus, from the most general down to the most specific. Jot that down. Most general to most specific. All members of the group have very certain characteristics that put them into that grouping. All members of the group have certain characteristics that put them into that grouping. And the groups have a name. We call them the taxon, or taxa for plural. Here is the classification system. Starts up here with domain, kingdom, phylum, class, order, family, genus, and then species. Yes, it is essential for you to know all of these in order. Domain, kingdom, phylum, class, order, family, genus, and species. You might complain, oh, I have to know all of those in order. Yes, but here's the actual classification system. Domain, kingdom, phylum, subphylum, superclass, class, subclass, cohort, superorder, order, suborder, superfamily, family, subfamily, tribe, genus, subgenus, species, and subspecies. And that's just for the animals. There are some other ones in there for the plants, so I'm not having you go through all of them. Just these main ones. Now there are lots of ways that you can learn these using the initials here, so I highlighted those. I just remember it: domain, kingdom, phylum, class, order, family, genus, species. If you want to have some other trick, you can look it up online to find that other trick. But you do need to know all of these. Domain, kingdom, phylum, and class is how we're going to be discussing these organisms as we move through the course the rest of the year. Now, what recent research has actually showed us some new developments. Because what we're doing recently now is we're looking at genetics. When we look at genetics and biochemicals like the proteins, we start to see very, very clearly those relationships between these different organisms. So what is developed from this research in DNA and genetics is what we have figured out is that there are 3 different domains. So of that domain, kingdom, phylum, class, order, family, genus, species, we have 3 domains. Within those 3 domains, there are 6 kingdoms. So in the picture here, the 3-domain system, we have the domain bacteria, the domain archaea, and the domain eukarya. So I wrote them down here. Our 3 domains: bacteria, archaea, and eukarya. Now you might see some of these are relatively familiar. Things like eukaryotes, those organisms are in the eukarya. So the kingdoms that go along with these domains, the domain bacteria has a kingdom called eubacteria. The domain Archaea has the kingdom called Archaebacteria, and the domain Eukarya, there's 4 kingdoms as a part of this one domain. So we're having this branching. The Protista, the Plantae, the Fungi, and the Animalia. Protista, Plantae, Fungi, and Animalia are in the Eukarya. Again, the Archaea has the Archaebacteria, and the bacteria domain has the Eubacteria. So these are the groupings. We're going to go through each one of these groupings in the course. We're going to start off in this unit with these 2 right here, the bacteria and the Archaea, of which there's only 1 kingdom in each one of these particular domains. This too is an essential skill, to know these 3 domains and the corresponding kingdoms that are within those particular domains. Now what we use these domains, kingdoms, phylums, class, order, family, genus, species, to do is to determine relationships between organisms. But it's very, very difficult. And it's difficult to determine relationships because when we try to determine relationships, we like to look into the past and see what those particular relationships are. And for most organisms, the fossil record is non-existent. So we have to use other evidence to find those relationships. Things like the structure of organisms. That's an easier one. We can look at them. How they reproduce. How they develop embryos and larvas, etc. Of course, DNA and RNA, proteins, that's the new frontier. Because this is very new stuff that we can actually look at the DNA of different types of organisms and see their relationships. And so what we do is we group these organisms, looking at that evidence, using that evidence, we group them to look at how they're related, so that relatedness part. In other words, how do 2 organisms share a common history? What is that evolutionary history that these organisms have? And we do this by putting these particular different types of organisms into a diagram called a cladogram. And this cladogram has these branches coming off here, you can see in red. And then there's some terms listed on here. The branches in red, that's where these different organisms belong. They belong on these particular branches. And then the words that follow along each branch, as you move from the base down here, the beginning, towards those organisms, are the characteristics that they have. So these organisms that are branched closer to each other, they share a lot more characteristics. There's only a few of these terms here that are different. The goal of determining these relationships is to see, well, what are the genes and how are they similar among these different types of organisms. And once we can do that, once we can figure out how these genes are similar, we can see their relationships and their common evolutionary history. Now, how do we construct a cladogram? And this one's a little bit more difficult to see. Couldn't change the black at all, but there's a few steps. So step number 1, choose an organism to look at. So that's our first step, to choose an organism to look at. Step number 2, determine which traits you're going to observe. What traits are you going to use to determine those relationships? Guarantee you, you're not going to be using all the traits. There are just way too many traits. Are you going to be using hair color? Is that an important one? You're going to be using how they reproduce? Is that an important one? How about their nervous system? Is that an important one? What particular traits are you going to be using? And then whether or not they have those particular traits. So number 1, choose an organism. Number 2, choose traits to look at and whether or not they're present. They have those traits. The third one is to group the organisms by those shared traits. So if the organisms over here have the trait, they're going to be grouped together. If all 3 of these have the trait, they're going to be grouped together. And then number 4, once you group these organisms, is to mark those traits on the cladogram. So put some markings here where they have those traits and label those particular traits so that all of the organisms above this point right here, that is this organism, and this organism, all are going to share that particular trait. The most important idea behind constructing one of these cladograms, one of these diagrams showing how these organisms are related, is to use the simplest diagram that explains all of the available facts. We call that the principle of parsimony, also known as Occam's razor. So the principle of parsimony used the simplest diagram to explain all of the available facts. And that's the end of the notes. Pretty quick and painful. We're going to be utilizing this information in our next class period to actually construct some of these cladograms, looking at the relationships that different types of organisms have.